All right, shall we get started?
We can, and can you hear me? This is Pat Caldwell.
Yeah, ma'am. Yes, I can.
Okay, good. Well, first of all, we'd like to welcome everybody on behalf of Collie Club of America Breed Education and the Collie Health Foundation. We're fortunate to have a Collie Health Foundation that does support our collies and try to give to them everything that we can. And since 1988, we have been, the foundation has been giving money, granting money for PRA research. And up to yesterday, I believe that amount is $194,666. And just recently, they have offered more grant money for CEI research. That's $47,000, looking for a modifying gene for that disease. And I do want to remind everybody about the work that the Health Foundation does do, and that they do offer rebates for the PRA testing. And I know that Sue is going to get into this and the work that's being done in behalf of our collies and other breeds too is amazing and wonderful, and it's something that we need to embrace and use for the betterment of our dogs. With that, I'd like to thank profusely both Sue Pierce-Kelly, who's doing this webinar, and Dr. Casey Knox, who will be doing, I think, a couple for us in weeks to come. Both women have graciously consented to do this in their busy schedules despite COVID-19 and all that everybody is going through, to offer this because we couldn't get together at our seminar. It is being recorded, I believe, and this will be available through both the Health Foundation website and CC of A Breed Ed. So a little bit, well, first of all, let me tell you that Dr. Casey Knox, both women work in conjunction with Wisdom Health, and Dr. Knox is the veterinary technical support analyst for Wisdom Health, and Sue Pierce-Kelly is the general manager that oversees, I believe, the testing that's done at Wisdom Health. Do I have that correct?
No, not quite. That used to be my position at Optogen, but when Optogen was acquired by Wisdom Health in 2018, then as we wound down operations at Optogen, I started working with Wisdom Health as an independent contractor. So I do whatever I can to assist, particularly in the breeder department and helping do some of the transfers of information and relationships, and especially the research that was going on at Optogen into the Wisdom Health community.
Thank you. You began early in your interest in your field. You had an English setter as a toddler, and then when you were young, you had a Brittany that succumbed to canine distemper virus. And that kind of brought you to your college degree. You were a biology major, I believe, and you got your bachelor's and then a master's of science in pathology from Cornell. And that's while you were working at James Baker Institute. There you were fortunate enough, in our mind, to be able to do research with Drs. and Dr. Ackland, and their focus was on multiple mutations involved in inheritable eye diseases in dogs. Two of those are, things that collie people are concerned with. That would be collie eye anomaly and the RCD2 PRA. In 98 Optogen, that's the testing laboratory in Ithaca, focused primarily on that, and then genetic tests through all that research has developed through that. You left Cornell as the manager and then told us what you're doing now. And you and your husband have lived for 40 years on an old farm near Ithaca, which is kind of wonderful. In 1980s, you showed and bred a few litters of golden retrievers. And one of my favorite remarks that you make is that you are raising your two sons, right, and you live there raising your two sons who are finally obedience trained. Perhaps you can write a book.
Yeah, for the most part, I think they are pretty well-trained young men. So, also, thank you, Pat. Thank you for the invitation, and thank you to the Collie Health Foundation and Club of America for this opportunity to speak to you about the inherited eye diseases and of particular interest to collie breeders. And I just want to make it clear right at the start that I am not a veterinarian. So I am very far from being a board certified veterinary ophthalmologist where all of this information really resides in that realm. But as you said, I had just a wonderful opportunity to work with Dr. Gus Aguirre and Greg Ackland from the early 90s. So at least by osmosis, I've acquired quite a bit of information from them. And many of these slides have come from them graciously offered to help me with the presentation. And any questions that you might have that I can't answer, I'll be sure to get in touch with them for further information. And as you mentioned, Collie eye anomaly and the RCD2 PRA are two mutations that Gus and Greg studied extensively. It took a long time to get to the finish line with those, but Gus and Greg were, just to give a little more background, were some of the pioneers, among a handful of pioneers in the early days of canine genetics. And in fact, it was Greg's boxer, Tasha, that had the in-depth genome sequencing done and then became the reference genome for continuing canine genetic research that carries on even today. They've always, both Gus and Greg have always been very grateful to the conscientious breeders and the breed clubs like the Collie Club of America, and especially the Collie Health Foundation that has supported much of their research. Without this kind of resource, these advances would never be made. So it's a really appreciated resource that you folks have provided to this. And of course, one of the main goals of their research is to identify the mutation so that a DNA test could be developed. And some of you may be, in the audience, may be new enough to the field of collie breeding to not know what it was like, particularly back around in the 70s when PRA was prevalent in the breed. I was trying to find a way to get the statistics on what the frequency was back there then, but from my understanding, it was the most important inherited disease problem in the breed. And for those in the old guard, they'll undoubtedly recall that the heartache of the early onset PRA, and the only way to determine if a potential mate was a carrier of the mutation was to do what was called test breeding. And that's where the potential mate would be bred to an affected dog to see if any of the offspring were also affected, I should say. With luck, no affected puppies would be produced, and it could be assumed pretty safely that the potential mate was indeed a non-carrier. And that was possible because this form of PRA is quite an early onset form of PRA compared to many other forms of PRA. Dogs are clinically diagnosed much younger than a year of age with RCD2 PRA. So it was very good information when you've got the potential mate to be identified as a non-carrier. But of course, there were dogs that proved to be carriers and in the test breedings, and that was important also to help curb the spread of the disease. But unfortunately, one that resulted in affected pups in order to obtain that information. So identifying the mutations for PRA particularly took many years and more than one frustrated graduate student, but finally all the hard work and the support from the Health Foundation paid off, and the test became available at Optogen in 2008. So that's a bit of my background, and hopefully provide you with a bit of context as to how we got to where we are today when it comes to understanding these diseases. And as Pat mentioned, there are two that I'll be talking about of particular interest to the Collie breeders, the RCD2 PRA and Collie eye anomaly. But before we get into those specifics, just to give some background, two tools that you have at your disposal as dog breeders are the eye exam and now, thankfully, the DNA testing. They're complementary tools. Neither replaces the other, and this was a message that we were continuously stressing at Optogen, because we would often hear people say, well, I don't need to do an eye exam because the dog had its DNA test done on pick a mutation, any mutation. And of course, they're not assessing the same thing. The eye exam is done by a board-certified veterinary ophthalmologist who examines the entire eye. And there are many conditions, inherited conditions in different breeds of dogs, including the Collie, that don't have a DNA test available yet. So the only way that you know if your dog is expressing one of these diseases is to have the eye exam done by a qualified professional like the ophthalmologist. And I would just like to stress that qualified professional assessment, because that's one of the differences between what happens in a DNA testing scenario versus the eye exam. Right now, the DNA testing for canine genetic diseases is completely unregulated. Anybody can get a PCR machine off the Craigslist and set it up in their garage and call themselves the best thing for canine genetics since Optogen or Wisdom Health. And in the eye exam, you've got a veterinary ophthalmologist who has done rigorous training and has the experience to pass judgment on the ocular health of your dog. These eye exams are typically done annually, and that's how the certification number, what we used to call the CERF number, expires 1 year after the clinical exam is done. And that's another difference between the DNA test. Of course, you can have your puppy tested as young as you can get a non-contaminated sample, and a blood sample, for example, from a week-old puppy, if you can get a veterinarian who would draw that, would be an indicator of the genetic makeup of the dog for its entire life. The genes don't change, but the ocular health of the eye can from year to year, of course, and some of these disorders don't show up as young puppies. Both Collie eye anomaly and RCD2 do show up very early on compared to many other diseases. And of course, the other advantage of a DNA test is it's able to detect whether the dog is carrying one copy of the mutation in an autosomal recessive disease, which I'll talk about shortly. And the eye exam cannot detect that. You have to have the genetic information in order to determine whether the dog is carrying one copy, and also whether it's carrying two copies of a disorder that may not be evident until later in the dog's life. This is the eye exam form that many of you are, I'm sure, familiar with, and it's typically done in triplicate. The one copy goes to the OFA, or used to be CERF. One copy stays with the veterinary ophthalmologist, and one copy is for you. And all of these different hieroglyphics in the middle are where the ophthalmologist can make comments on the left eye and the right eye, and they serve to provide ongoing statistical information on the frequency of any of these conditions that are listed on the form on a breed-by-breed basis. There are currently 10 conditions that prevent eligibility for an OFA or CERF number, and they're listed here. I bolded optic nerve coloboma and PRA because those are two disorders of particular concern for the collie, as they have occurred at high frequency over time. And some of these others that have asterisks just should be noted, they are not necessarily inherited, and can, if there's a clear cause, traumatic cause or acquired reason that the condition exists, it wouldn't preclude the dog from being bred. So going on to Collie eye anomaly in the eye exam, as I said, it's performed by a board-certified veterinary ophthalmologist, and it's this particular condition is congenital. It's there at birth, and it's best observed when the pups are very young, 6 to 8 weeks of age. And that's because as the pups age, the dogs age, this choroidal hypoplasia, which means a lack of development of this vascular tissue of the eye, will get covered up as other layers, pigmented layers of the back of the eye grow over it. So the ophthalmologist can't see that the hypoplasia is there. It's there, but it gets covered up like a curtain being drawn as the pigmented cells grow over it. And it's characterized, Collie eye anomaly is characterized, I think in all cases, by this hypoplasia, this whitish area in the upper part of the eye, typically in the temporal or the side near the side of the head. And typically, almost always, it's a bilateral observation of choroidal hypoplasia. It's in both eyes. And the good news is that in and of itself, the hypoplasia does not appear to have any impact on vision. The problem is when other clinical signs that often, well, occasionally now I would say, that sometimes accompany the CEA occur, and those do have the potential for significant impact. And particularly it's the optic nerve or optic disc coloboma. I'm sorry? Nope. Everything going okay? Everybody can hear me?
Yes.
Okay, great. I heard somebody say something. I wanted to make sure I wasn't going
I think
off.
someone joined without muting. If they do want to ask a question, feel free to unmute and ask your question.
Great, yeah. So on the left, I love this picture of the normal fundus of a dog. It reminds me of that space shot of the Earth back when I was growing up, but here's the optic disc, and this is where the axons from the ganglion cells, those are in the layer of nerve cells in the retina, they all go through the optic disc back to the brain, to the central nervous system, to relay the information from the retina. And this main ciliary artery here on a normal dog, you can see is straight and shoots off like a branch.
The volume back in the house attached to the end of the
When you have a dog affected with Collie eye anomaly, you can see how squiggly and tortuous these blood vessels can become. And then it's hard to see on this photo, I have some other ones, but there's a little circle here at the optic disc, which is the optic disc coloboma. And a coloboma is basically a pit where the development has been abnormal. And here's some more obvious views of one. You can see where this outline here of this grayish area, and that is a fairly large coloboma in the optic nerve, and it's got to have some impact, I would think, on vision in this dog. And the other problem, and here's another one, the even bigger problem than just on the optic disc, is when these colobomas get large, they can lead to retinal detachment, and that almost always results in that eye being blind. So the information that the ophthalmologist gathers and sends into the OFA is collated and collected, and then it used to be every year or two, they would put out an enormous, like 3-inch, very heavy book called the Blue Book. And it had a breed by breed description, multiple pages of descriptions of what the current observations were for various eye diseases in that breed. And here's the most recent one for the Collie, showing one of the pages that discusses or provides to the ophthalmologist recommendations on how they should recommend to the breeders when they observe a dog with any of these conditions. And there are 2 basic categories when they observe an abnormality, an ocular abnormality, either breeder option, which is the up to the breeder whether they think it's significant, like dystichiasis is multiple eyelashes, and when there are too many multiple eyelashes, it can be a bit of a problem, but it's nothing compared to, say, progressive retinal atrophy, where there's a no. Now, interestingly, and you'll see why this is interesting in a few slides, the Collie eye anomaly is also, or choroidal hypoplasia is also recommended as a no, but I think it's primarily when they recommend that when these more severe abnormalities occur. Just the choroidal hypoplasia by itself, in speaking with Gus about this, he said there's no way that in itself should preclude a Collie from breeding. And particularly in light that there's a bit of catching up that needs to happen in some of these recommendations. And in some of the pages of the Blue Book, you will see the advice that the DNA tests exist and dogs should be mated only to a dog that's been tested and clear of those mutations if they're showing the clinical signs. And here are some of the statistics from the Blue Book that are just, to me, just quite amazing how common Collie eye anomaly is in the Collie, particularly the rough Collie. I'll show you some genetic information on that shortly. But here, there's 2 different time brackets that are in the Blue Book. And from 91 through 2013, about 68% of the Collies examined were diagnosed with choroidal hyperplasia, and about 4% were diagnosed with optic nerve coloboma. That number is interesting in its change over the most recent few years, that it's gone up to close to 75% of Collies that are diagnosed with choroidal hypoplasia, and the number has dropped in coloboma. I can say that this 0% is not completely accurate. It's just below, it's less than 1%. We certainly do see Collies with coloboma, and it's just fortunate that the numbers are dropping, and we'll talk about that shortly as to why that may be happening. Happily, the frequency of PRA in Collies now is rare, particularly compared to where it was in the 70s. And again, this 0% for the most recent is not accurate. It should be less than 1%, because we do see Collies that are affected with PRA, including the RCD2 PRA. But I should mention that one of the other reasons to continue to do eye exams is just because we have a great handle now on the RCD2 form of PRA, there is absolutely no reason that another form of PRA cannot occur in the Collie. I would say over time we will probably find that the majority of dog breeds have more, there's more than one form of PRA that is observed, and in most cases, not all of the forms of PRA will be known. So that's one more reason to continue to do those eye exams.
Sue, this is Pat Caldwell. I have a question, or maybe a comment on this. When we do our PRA test now, and we get the when we do our Optogen tests, there is a PRA test that's on there when we do that broad scan of it that shows MDR1, coat type, all that. That is not Collie PRA that is on there. The PRA test that people need to be doing is the separate one that they have to order separately from that on Optogen, correct?
That is correct. Thank you, Pat. And I'll mention this again later, but that's exactly right. So the RCD2 PRA test is not part of the optimal selection panel, and that's because it's special. It requires extra steps in order to accurately detect the mutation. It requires a step called demethylation of the DNA that none of the other mutation tests require. So for that reason, it's just technically, it doesn't play nice on the panel. I think Wisdom Health is looking at ways to perhaps have multiple redundant markers that would be in what's called linkage disequilibrium with the mutation that would allow that kind of a test of markers that would strongly indicate whether the PRA mutation is present, to put those markers onto the panel. But I don't think it's quite ready for that commercial use, but I know they're working on that so that you would have a very strong indication, very tightly linked markers that could be run on the panel instead of having to do the separate mutation-directed test.
Right, thanks. I just wanted to point that out early so that when you say it again, people will have heard it a couple times. And the other thing, I know that when I'm talking about doing the PRA tests and I'm talking about doing a breeding and I'm looking at my pedigree and I talk to stud owners, one of the first things I ask is, what is the PRA status on your dog? And I have had some people say to me, we haven't had PRA in our dogs for generations. And it's like, well, what's the proof? Well, I tested, the dog like 4 generations back, and it's like, that's not how it works, just because of what you said about the mutation. So they do the PRA test for both parents. The puppies are now PRA clear. What happens next?
That's a really good point, Pat. The recommendation that Optogen always provided was there's really nothing that can replace the actual testing of the dog itself. And that's because errors can happen all along the track, and particularly with sample collection. If the wrong dog is collected, or if the wrong sire gets over the fence and that dog is a carrier, then you've got puppies that, unbeknownst to you, are carrying the mutation. And next generation, when you breed those puppies thinking they're clear and they're not, then you wind up with the disease there all over again, and have, it's sort of like coronavirus. The more testing and monitoring you can do, the better you're going to be able to prevent a bad outcome. And various registries have different rules that they use. I think the OFA currently requires parentage testing and will allow 1 generation not tested, but you have to do the parentage testing in order for the dogs to be registered on the OFA as clear by parentage. So to my mind, you're just as well off to do the direct test itself. And hopefully that test will be available on the panel, so it'll just be automatically as a strong marker-based test, and it'll just come with the rest of the optimal selection results. And when you look at those optimal selection results, and I'm glad you brought this up, you will see a whole plethora of other forms of PRA. It'll be in a different section of the report. The report will segregate out to the top of the report any of the disorders that are known to be of particular concern to the Collie breed, and then there will be more than 100 other disorders that are also tested. And this is a really wonderful survey opportunity, screening opportunity to make sure that one of these other mutations that might be present in a rare portion of the breed isn't there and doesn't crop up at a higher frequency because you didn't know it was there.
Sue,
Mhm.
Timer is here saying you're at halftime.
Thank you. I better hurry up. um, the research I mentioned earlier is being done at Dr. Guerri's lab on optic nerve colobomas. So I really encourage anyone who has a collie that's been diagnosed with an optic nerve coloboma to contact the folks in Gus's lab, and this information will be available, of course, with the webinar, and hopefully they can make progress on identifying what other gene or genes are working in combination with CEA to get to cause the colobomas. And as we've mentioned, these two mutations for CEA and RCD2 were identified back in the mid-2000s by Gus's group, and CEA was also, research was done in combination with a wonderful, another pioneer of canine genomics, Lane Ostrander's group, and Heidi Parker, I believe, did her PhD on CEA and CH. And Anna Kukakova, who also worked on the CEA, and Orly Goldstein in Gus's lab found the RCD2 mutation in 2008, or in 2009, and or published it then, I should say.
So we have a question from the chat box.
Okay.
Someone asking about differentiation of colobomas. For example, if you have one in the right optic nerve, what would be the implications of some of those varying severities or locations of the colobomas that you mentioned before?
It's a good question. Interestingly, the colobomas, unlike the choroidal hypoplasia, which is almost uniformly bilateral, the colobomas are less consistent in that regard. So does that get to the question that you can often have a unilateral optic nerve coloboma? Does that answer the question?
I think it's more like the location and the size and things like that. Is there any implications in terms of prognosis? And Katrina, correct me if I'm wrong in overinterpreting your question. Yeah.
Definitely there is correlation. The larger the coloboma, the greater the risk of retinal detachment. There's probably a complex developmental pathway that's involved in the development of these colobomas, but hopefully the research can at least point to some of the main genetic factors and begin to be able to control or at least understand them better once the genes are identified that are involved in it. It's also a problem in humans.
If I could just ask, this is Katrina, if I could just ask to clarify. So my question was, does a coloboma occur, can a coloboma occur in multiple areas of the eye? For example, the iris, the optic nerve, other areas, because it's a pit, if I'm understanding correctly. And if that's the case, then are there different implications for where the coloboma occurs?
Yeah, that's a really good question, Katrina. Yes, I should have clarified that. The colobomas associated with Collie eye anomaly are the optic disc colobomas. But there are other colobomas you might have heard of, for example, the iris colobomas, and you'll see those often in Merle dogs. And the Merle gene.
What kind of dogs? What kind of dogs?
The Merle-coated dogs.
Okay.
Because, and actually it's another point about Merle, it affects the distribution of the melanin in various tissues, and it can be associated with other ocular abnormalities like these colobomas of the iris. And one of the things, corollaries that we've seen at Optogen is there occasionally will be clients who will get in touch with us who say, how can my dog have choroidal hypoplasia when it's clear of the CEA mutation? And I think virtually all of those cases that we would hear about were in Merle-coated dogs. And the look of choroidal hypoplasia can be very similar to the look of a Merle-coated dog when the ophthalmologist looks at the fundus, particularly in a squiggly, wiggly little 6-week-old puppy, it's hard to get a good view in those little almond-shaped eyes of what the back of the eye looks like. So occasionally CEA will be misdiagnosed when it's really the Merle coat pattern that's causing the appearance.
Okay. Thank you so much. I appreciate that.
Sure. Moving on to some genetic testing information on CEA and RCD2. This is from the last, up through the last of the tests that were done at Optogen before we transferred all the testing over to the Wisdom Health Laboratory. And you can see here, it's just, to me, it's just amazing how high the frequency, and these are dogs that are homozygous for the CEA mutation in the most recent batch or category from 2014 to 2019. Half of the rough collies that were tested at Optogen carried 2 copies of the mutation, and 35% of them were carriers. And there's the minority of collies, of the rough collies, and of any of these collies, were clear of the mutation. So thankfully, it's a mild condition, and I'll talk a little bit more about that shortly, because it didn't necessarily used to be that way. And then again, thankfully, the incidence of the RCD2-PRA mutation is low. Had we had this kind of information back in the 70s and 80s, these numbers would be much, much higher. But between the combination of test breeding and vigorous use of the genetic testing, the incidence has gone down. And I'd like to point out a really wonderful relatively new website. It's called mybreeddata.com. I love this website, and it's supported by the Wisdom Health group and their research team. And it's all the data are live, and they're generated from the optimal selection test results. And it allows you to go to the site and search either by breed and see what tests are on the optimal selection panel and the frequency of those mutations in any particular breed, or you can search by disorder and see what other breeds carry that particular disorder and the frequency of the mutation in those other breeds. For example, Collie eye anomaly is observed in many different breeds, most of which are of Collie relationship. I don't know about that Hokkaido dog. I always wonder where Collie eye anomaly got onto a little island in Japan, but I think maybe in World War II, somebody brought a border collie with them or something to the island on the ship, and it got into the Hokkaido dog. But again, the frequency of collies that genetically at risk is what Optogen used to call affected, but this is a better term in many ways because affected really should be relegated to a clinical diagnosis. So genetically at risk indicates that the dogs are homozygous or have 2 copies of the mutation. And look at those numbers. It's just amazing how frequent it is. And why is there such a high frequency of it in the collie? And again, the hypoplasia of the choroid by itself is not a major concern. So there's been no strong selection against that particular clinical sign. And my guess is that the loose association between the Collie eye mutation and the more severe conditions of optic nerve coloboma and retinal detachment have allowed the breeders to begin to segregate, to a limited extent at least, the mutation for CEA away from the genes involved in colobomas by selecting the dogs or lines where the colobomas don't occur. And Greg Ackland had shared that this type of modification of the CEA severity through selective breeding has occurred before, that many of the ACVO diplomats encouraged the collie owners to breed the least affected dogs together years and years ago. And this is what Greg concludes has resulted in this go normal phenotype of a very mild hypoplasia that doesn't, for the most part, involve the more severe conditions. And there's one misconception that I want to try to make clear. And some of the folks I've talked to over the years in the Collie groups have thought that that Collie eye anomaly itself was the cause of that lovely almond shape that the Collies have in their eyes. And this is clearly not the case, as Greg has said to me, that they very early on in the research colony, they bred the Collie eye affected collies to other breeds of dogs that had larger eyes, because it's such a bear to try to get a definitive diagnosis on small puppy eyes. And so they were within 3 generations able to put the Collie eye disease or phenotype into a breed with much larger eyes where they could do the diagnosis much better, and clearly segregated the eye shape in the Collie from the mutation for the disease. And many of you are, I'm sure, familiar with the CHIC program, and the RCD2 test is part of CHIC, but the CEA is not. And that RCD2 test, as Pat noted, is available on what used to be the Optogen website. It got transferred over to, or redirects over to this site here, the breeder.wisdom panel, and that's where you can find both the RCD2 test and also the DMS, the dermatomyositis test. I'm not going to talk about that because it's not an eye disease, but I think Casey will be talking about that in a future discussion. And then the other tests that you want to have done for your Collie for CHIC, the multiple drug sensitivity, the MDR1, that is available on the optimal selection panel, as is the Collie eye anomaly. Currently, the Collie eye anomaly test is about a $20 add-on. Hopefully, in a short time, we'll get the licensing reconfigured with Cornell, who has the patent that is exclusively licensed to Wisdom Health for CEA. We'll get that worked out so that it'll just be part of the panel without having to do the add-on step. And working on the basic steps of DNA testing. Whoops, go back. As many of you are, I'm sure, familiar, the basic steps are you collect the DNA from pick a tissue, typically whole blood, or most commonly now the cheek cells with a swab, but you can also use the sperm cells and semen.
So can I interject for a moment?
Yep.
You've got 15 minutes left, and we have a couple questions for you.
Okay.
Kathy, I think you'll probably have to pose your question yourself, because I'm not quite sure what you are wanting to know. But then Katrina also had a question about how close we are to gene therapy for treating affected dogs with Collie eye.
Okay, gene therapy. Oh boy, one of my favorite topics, and a whole hour or more of discussion itself. And Gus's group, I would love to talk more about this, but it was his group and Greg's group that developed the first gene therapy for dogs in a type of disease called congenital stationary night blindness that occurs in the Briard. And the exciting thing about that research is that it led to the first gene therapy for children who have that very condition. It's called labor congenital amaurosis, and it's now a clinical treatment that all began with the basic research of finding the mutation that occurred in Briards, and then doing gene therapy treatment on the Briards. And I remember the day when the gal in the kennel said, "I think Lancelot," that was the name of the Briard who had one eye treated with this gene therapy. She said, "I think Lancelot can see because he's following me around through the window. He's watching me." And sure enough, he had vision in the one eye that was treated. And anyway, regarding other forms of gene therapy, right now there are no gene therapy treatments for Collie eye anomaly or any of the PRAs right now, but there are some gene therapy treatments being done on a type of color blindness, really, or day blindness, it's very exciting to see that work coming out of Dr. Peterson-Jones and in University of Michigan. In order to develop gene therapy, it takes a commitment and some effort. And given how mild the Collie the hypoplasia is, it probably would not be worth the effort just for the hypoplasia itself. But it's a fascinating area to look into. So just finishing up the DNA basic steps. Once you get the DNA, you extract it, or you get the extracted DNA and you amplify it, and then you select, or you send in a probe that's able to detect a region of the DNA very close to surrounding the mutation. And there are many different ways and many different types of probes to do this. Some work better than others. And the whole goal of that is to have the probe determine whether there are 0, 1, or 2 copies of the particular mutation you're looking for present in the sample. And a variety of things once you've got that DNA test information available on how do you use the information. My go-to is the breed club or health registry recommendations, because those are the folks that know the breed the best and should be able to advise breeders on how critical a particular disease is and how to handle it within the breed. Something like CEA, you've got to be very careful before you get heavy-handed with trying to get rid of CEA in the Collie given its frequency. Versus, say, RCD2, it could be a decision made within a club that, okay, let's try to eradicate RCD2, and you're doing a pretty darn good job of doing that. But the correlation of the mutation with the disease is an important factor, not so much in these two diseases that we're talking about, but there are some other DNA tests where the mutation doesn't have, it's maybe you would call it, it's a low penetrance. When the mutation is present, it doesn't necessarily mean that the dog is going to develop the disorder. But other than the Coloboma issue with CEA, that isn't the case with these two diseases. The mode of inheritance is an important factor. I'll mention that in a minute in determining how to use the test. And of course, the prevalence of the mutation, as we just discussed, and the severity of the disease. All these are things that guide a breed club in making decisions on how to best control the, or a breeder, how to best control the incidence of a disease using the genetic information. The mode of inheritance is simply how that trait or disease is passed on from one generation to the next. And both of these conditions, RCD2 and CEA, could be described as autosomal recessive. RCD2 definitely, CEA you could describe as complex because of the associated coloboma issue, but the choroidal hypoplasia itself is a simple autosomal recessive trait or disorder. There are other modes of inheritance that we see in inherited eye diseases in dogs. There's an autosomal dominant form of PRA that occurs in mastiffs. And that means that all it takes is one copy of the mutation for the disease to occur. So that pretty much precludes that dog from a breeding program because it has a 50-50 chance, if it's got one copy of the mutation, of passing it on to any of its puppies. And then there are sex-linked or X-linked recessive forms of PRA. We see that in the Samoyed and the Siberian Husky, where the mutation is on the X chromosome. So typically you see the disease in males, as the females tend to be only mildly affected, if affected at all, but when they carry one copy of the mutation. But if they pass that X chromosome on to their males that carries the mutation, the males will show disease at a fairly young age. So moving on, and most of you are probably familiar with this Punnett square type of how you use the genetic information to make your breeding plans. And typically what we're shooting for is avoiding any dogs that are affected by breeding the normals to anybody, and the carriers of either 1 or 2 copies of the mutation only to a normal. So in most cases, I'm saying most cases because again, chordal hypoplasia is such a mild condition right now that I think it's an exception that you may not need to avoid producing an affected offspring, but the problem is there's always the chance of those Colobomas until we have a better understanding of the condition. But in general, the rule of thumb is you want to have at least 1 DNA tested normal clear parent to be a part of a breeding pair in regards to an autosomal recessive mode of inheritance. And in general, you want to be, how do you use the DNA test results yourself as a breeder, be as well as informed as you can, and particularly on the laboratories that are doing the testing, and use your best judgment. You want to think of these DNA test results when they come back as a carrier of a disease as a flaw, and it's just like in a confirmation, or a fault, I should say. But it's not the whole dog, and you don't want to throw the babies out with the bathwater just because the dog's carrying a mutation or 2. We all carry many mutations, and the great value of the DNA testing is you can still breed those dogs and avoid the disease, and while keeping all of the good traits in the dog and the line going, but avoiding the disorder. And finally, the question of where to test. And it's an important question because, like I said, there is a whole spectrum of laboratory quality out there, from some pretty bad players still on the scene, unfortunately, to some excellent ones. And it's difficult to know how to decide. And there's a group that has gotten together somewhat recently called the International Partnership for Dogs, whose aim initially was really aimed at trying to make more transparency and help people assess where they should send their samples for testing. And some of the factors that I think are important are listed there. Intellectual property, where it exists, and very few of these tests have patents, but some do, including CEA. And there's a whole other hour or more of discussion on DNA patents and the legal area surrounding them. But at the very least, one thing that nobody can dispute is that the royalties that they generate by when laboratories license these patents goes back to the research institutions, and that allows a continued support for ongoing research, which everybody wants to see happen, so we can continue to make progress in finding new mutations and developing new tests to improve the quality of health for our dogs. And then the reputation of the laboratory, does it have a good track record? How does it handle errors when they occur? Some laboratories do nothing and say, tough luck, Charlie. Others will put a full investigatory effort into it to understand whether the error arose in their hands or whether it was a sampling error, and that makes all the difference. Errors happen typically at the sampling level, but it's important to a laboratory that cares about this to know where any error might have arisen. And that leads to the quality control point, which probably should be number 1. And in that regard, I think there's one gold standard in laboratory quality assessment, and that's the ISO 17025 accreditation. It's an international set of standards, and it is a bear. It's hard to achieve. We were very, very proud at Optogen to get it when we set our minds to it after, I think, 2 years to get accreditation, and then from 2014 on, we maintained it. And I'm very happy to say that the laboratory that Wisdom Health contracts with, that's GeneSeek here in North America, is also ISO 17025 accredited. You won't find a lot of canine genetics labs that are. Professional expertise, do they have veterinarians on board like Dr. Knox? And there are a plethora of excellent veterinarians in the Wisdom Health group, as well as PhDs in canine genetics. And then finally, expanding understanding, do they support ongoing research? And I'm really happy that the legacy that Optogen had of of continuing research is very much at home in the Wisdom Health group, because they have a really robust research program and are continually identifying new mutations and putting those to good use in DNA testing. So hopefully I haven't run over time.
You have 2 minutes to go, so we can open it up for questions.
And a thunderstorm is just moving into my area, so hopefully we'll still have power in the next 2 minutes. But I didn't get to one question I think that somebody had that raised their hand.
Yeah, I think it was surrounding the variability in Collie anomaly expression. And so the comment was affected is affected, right? But I mean, do you want to speak a little bit to the variability that we see in expression of it beyond just the go normal phenomenon?
So the variability of, well, the degree of hypoplasia can be quite variable. You can have just a mildly affected lack of development versus a huge patch of the upper eye completely lacking a choroid. You could see, I think, in those fundus photos with the eye vessels, how some of them had just amazingly tortuous vessels like this one. Whatever is causing this, the complexity of the development of colobomas and the degree of hypoplasia is probably going to be an interesting developmental pathway to understand, because the breeders themselves have done a great job in reducing the severity of a lot of these variables just by breeding the dogs that have the least severe signs. Like this area here is all hypoplastic in the choroid versus up here where you can see the tapetum clearly. But that's a pretty big area of the upper eye. This is maybe maybe a third of the upper eye is affected versus this area here which is much smaller. So again, it's not known what causes the variation, but I'm hopeful that the research that Gus's group is doing in identifying the coloboma will begin to shed some light on that.
Sue?
Uh-huh.
This is Pat Caldwell. I'm not sure you answered Laura, and I think it's Matobi's question. She wanted to know what accounts for the difference in roughs and smooths on that chart that you had.
That is a good question. I don't know. I really don't know. Let's go back to that.
We kind of touched on it in the chat a little bit. A lot of these breeds, because there is a semi-separation of the breeding populations, you can have different frequencies. Same thing is true if you look at Collie frequencies or Sheltie frequencies for diseases or even traits. Even things like the immune system type that we see for DMS can differ by country, having to do with the populations that exist there. In addition, it's reflected, what those data are the tested dogs data. So if we happen to get a certain subpopulation of rough or smooth collies that have done testing, we might see some differences there. And then sometimes we have people that have questions about why there's a difference in the frequency overall that we report, because sometimes there's a pretty large magnitude of difference compared to, say, what was posted on OFA. And one thing I like to remind people is, as Sue mentioned, everyone carries genetic mutations, but there's still a bit of a stigma sometimes about that people have about posting on OFA for certain disorders. And so these frequencies are actually all of the dogs that have been tested with us, and then the information has been anonymized. So there isn't any what they call reporting bias for it. So, and then this is also going to be dogs that are tested through My Dog DNA, which is primarily our European and Asian purebred dogs tested with us.
Okay, so Laura did have a question with what you were saying. Her comment is, but roughs and smooths tend to be interbred, so wouldn't that even out the status?
They are interbred, but it's not a perfect diffusion of genes. So any breed is not going to be a perfect homogeneous bundle of dogs, right? So you're going to have little pockets of things here and there, just like you have certain characteristics by line. And so you may have certain lines whose genes have been magnified in the population. Say there was a popular sire and he was or was not a carrier for something, that will affect what you see within that population. It's not always going to be an even distribution, just because as breeders, we actually have a pretty strong influence over what's in the population. So those choices are going to impact what ends up in those smooth or roughs or British or American collies. And that's what those figures are.
Yeah, it's too bad that the OFA or the ophthalmologists don't distinguish in their clinical diagnosis between the rough and the smooth. They're combined together. It'd be interesting to see if the pattern, I would expect the pattern does hold, that the roughs have a higher percentage than the smooths, but yeah, it's a good question.
So we're over time by 5 minutes. I appreciate all the people that stuck around. We can stay a little bit longer, I think, and answer questions for those who want to stay, but I did want to let everyone know that if you have follow-up questions, and I know for myself, I often think of the question after the webinar is over, that Pat has graciously agreed to forward on and collate the questions that come in after the fact. If you email Collie breededucation@gmail.com, it's also in the chat box. And we'll be providing a recording of this webinar and then some of those resources, online resources that Sue mentioned for those that want to go look those up. And then, of course, if you have any questions, Sue and myself are both on the Wisdom Health team to specifically support breeders, so you're welcome to reach out to us with any questions as well.
Thank you.
I'd like to thank you on behalf of everybody who was here and everybody who has been emailing me asking if this would be recorded, because they're very excited about it. And I think what that says is that we have a support team out there who are professionals trying to help us do the best for our collies and dogs in general. And we're most appreciative of your time and everything that you've done for us. Thank you very much.
It was a pleasure to help, and we hope to help more down the road with further research.
Thank you so much. Take care, everybody.
Take care. Bye-bye.
Take care. Bye-bye.